Gabriel Victora with you guys, with us today, sorry. And Gabriel is a native of Brazil, and where he also had his undergraduate studies. And then he moved to the US at Rockefeller for the PhD, and then his independent career at the Whitehead Institute of MIT, and back to the Rockefeller more recently. Gabriel's interests are revolving around the germinal center reaction. It is, of course, key for affinity maturation and class switch recombination, and definitely the production of the most effective antibodies that we can. And his contributions in the field have been no short than outstanding. And I will just select two for you today to meditate about. One of them is his first paper as a graduate student when he was with Mike Nussensweig at Rockefeller. And basically, he tried to solve the long-standing issues of the dynamics of B cells migrating from the light zone to the dark zone and vice versa in germinal center cells. So basically, that was a problem that many scientists, including my former boss, I have to say, tried to solve by looking, at minute time scale. And then Gabriel came out and gave a major contribution just by doing something that is technically difficult, but conceptually very smart, which is changing the time scale from looking at, at events that take place in minutes to events that take place in days. And this resulted in a fantastic Cell paper back in 2010 that was the start of his scientific career. So when he graduated with that work from Mike Nussensweig lab, he was so good that it was actually not needed for him to do a postdoc like almost everyone else. He was directly recruited to the Whitehead Institute of the MIT and got an NIH Director's Early Independent Award for that. And the second piece of news that I wanted to tell you about that speaks extremely highly of Gabriel as a scientist and a lab leader is that he is definitely the only one I know whose undergraduate student had the opportunity of publishing a first author paper in science. So no, not a PhD student, but one of his undergrads, and which is definitely something that you don't see every day. All right, with that said, I leave the stage to you, Gabriel. Again, thanks to be with us, and looking forward to hear your presentation.
Thank you, Francesco. This was the kindest introduction I've ever had. Thank you. Let me see if I can get this right and share the right screen. This should be the PowerPoint. Now let me give it play. Can you, are you seeing the main screen or the presenter screen?
Main.
Okay, thank you. So thanks again. This is great. I wish I were there, but since I'm not, I'm imagining that I'm there near the beach, looking at the sunset or something. And I'll tell you a little bit about some of the work, actually, the work that Jeroen did in the lab that Francesco was mentioning, and some other, and how that turned into a whole area for us of this thing we call now clonal dynamics, and how clones live and die in the germinal center and become these high affinity antibodies that will hopefully protect us against COVID soon. I'm going to start with something simple, just a little bit of background. Everybody knows this, but I like to show this as sort of to understand our motivations to why we study clonal dynamics. And this is a picture that Linus Pauling drew in 1940, which was 25 years before we knew what a B cell was. But people had a pretty good idea of what antibodies did at that time. And that generated what probably was one of the founding questions of molecular immunology and cellular immunology as we know it, which is sort of what I call this original antibody puzzle. It is how is it that you can make antibodies that have high affinity and high specificity against almost anything, as mice and humans can do. So this drawing, I like it because it summarizes everything that we need to know to talk about this in one picture. This is a hapten here attached to a protein, and it's being hugged really tightly by this antibody that this was before anybody knew what an antibody looked like. But what is pictured here in this tight hugging is that this has high affinity, which is the key thing that an antibody needs to do. It needs to latch onto something and not let go, right? The other thing that's represented here is this narrow specificity. There is this minus charge here and the plus charge that complements it in the antibody. And if this hapten here had a positive charge over here instead of a negative charge, you wouldn't be able to bind it with this antibody. You'd need another antibody, right? So this is basically saying antibodies bind very specifically to things, and for every different thing you have, you need a different antibody, which leads to the third part of the puzzle, which is you need unlimited diversity. You need a huge number of different antibodies to bind a huge number of different antigens that you could present the system with. In the many years since that, we've made a lot of progress in understanding this. The part everybody knows about is that antibodies are not single genes. They're assembled during B cell development in a recombinatory way, and so that the 6 antibody genes that we have can create millions of different antibody binding regions here at the tip of the antibody. And that takes care of some of the diversity and some of the affinity, but there's this second part that people don't usually think about so much, maybe more now with all of the viral immunology that's happening, is that this initial rearrangement is not enough to lead to high affinity. Affinity seems to be something that's learned progressively during the response, and this was the original work that showed this, quantitatively at least, was from Hermann Eisen, who was at WashU at the time, and then later was at MIT for many years. So I had the chance to interact with him quite a bit when I was there. And what Hermann showed was that the response to an epitope, in this case a hapten nitrophenol, is not fixed. It evolves over time. So if you just look at the mean affinity of the antibody in serum in an animal right after immunization, it's not the same as you'll get several weeks later, meaning that the immune system is learning how to recognize an antigen better and better over time, right? And this is not necessarily a consequence or necessarily predicted by this type of rearrangement here that happens during B cell development before the cell even sees antigen. So in the 50 or so years since Hermann's paper, we have come up with a pretty good framework of how this works, with sort of a cellular framework for this improvement in affinity, or for the generation of high affinity specific antibodies. And basically what this says is that when antigen comes into the system, it meets the product of the VDJ recombination reaction, which is a repertoire of B cells. The huge majority of these B cells cannot bind to that antigen, but some of them can. And when the antigen comes in, it selects out the cells that can do this. And I kind of color coded this here. So the antigen is red. It won't find an exact match, but it can find this sort of red cell, and that's enough. It will trigger that cell to go into the game and start the reaction, right? Two things happen when this first cell gets recruited. One thing is some of the descendants of the cell immediately start to make antibody, and it's not very good antibody, and that's the initial antibody that was measured in Hermann Eisen's graph there at 2 weeks. But the most amazing part of this is what happens to the cells that don't become antibody producers. They start proliferating very vigorously. They go into this reaction that we call the germinal center. And there, they start expressing this enzyme AID, which is a mutator enzyme that everyone who works on B cells knows very well. And one of the things that this enzyme does is it introduces point mutations into the antigen binding part of the antibody gene. And these mutations are random. They have nothing to do with the antigen. So you end up with a panel of B cells that are randomly mutated. A lot of them are bind not as well to the antigen as the original cells did bind, but some of them might bind better. And occasionally you have a cell that binds actually really well. So then, because this antigen is still retained in the germinal center, you can select this cell and selectively expand it in the population. And this is sort of a very Darwinian framework. You have all the things you need that Darwin talked about, which is descent with modification right here, meaning you make a varied panel of descendants from your original cell, and then you have fitness-based selection here. And if you do this over and over, you enrich for the affinity in the population here. And then this can give origin to high affinity plasma cells that at 8 weeks secrete that high affinity antibody that you see in the homogenizing diagram, right? And this has been described as Darwinian many times. This is one of the common phrases, Darwin in miniature. This is a more poetic one. I like this one a lot from a friend of mine, ex-Nussenzweig alumnus, Vasco Barreto, Nature's Most Accomplished Portrait. As in, nature evolved to make a little mini evolution inside it that allows us to fight rapidly evolving viruses. But this picture, it's correct. We know that a lot of these things really do happen, but it is very incomplete in that it tells us that we are evolving a whole system to respond to a single antigen that's monomorphic, right? And we know that that's not quite what the immune system does. So for example, the homogenizing picture I showed you was against a hapten. This is also a hapten. We know that the immune system really likes these little hapten molecules. And if you take all these little haptens and you tag them onto ovalbumin, the immune system will tend to bind to the hapten and not to the carrier, because the immune system really likes this hapten. So it means there's another level of complexity here, which is how is it that the immune system chooses what exactly it wants to target? If it wants to target this little thing here, or if it wants to target this big surface here, and what are the rules that play in there? And this is what I'm gonna try to start talking about as a problem and something we have as a long-term goal to solve. And the reason why this is important, I think at least one of the reasons, is that not every epitope is created equal. For example, if you look at flu, there are some great broadly neutralizing antibodies that have been characterized over time by many people. They tend to bind, for example, the flu stem, or some of the HIV broadly neutralizing antibodies tend to be able to sneak into the CD4 binding site. And what's common about both of these types of antibodies is that they're targeting epitopes that the immune system usually doesn't like. These are non-immunodominant epitopes. And at the same time, they have very, very high affinity, meaning that they have gone through the germinal center reactions, sometimes as in the case of HIV antibodies, very, very extensively. And this creates sort of a paradox, right? If the immune system doesn't like these antibodies, how can it keep them in the germinal center for long enough to get their affinity to be that high that they are useful? And maybe this has to do with why it's so hard for people to generate these antibodies naturally, right? So I'm going to extend the evolutionary analogy a little bit more. Maybe this will sound a little bit silly, but I think it makes the important point that I want to make, which is the cartoon that I showed you before looked something like this. When you're thinking about intraclonal competition within the same clone, it very much is a selection for the fittest in a way that we can understand. So sort of, you know, the giraffe with the longer neck is a little bit taller, so it can eat more leaves than the one with a shorter neck. So this one's gonna survive, the other one is not gonna survive. And that's conceptually very simple to understand. But what we really are looking at when we look at a germinal center might be something much more like this. There's a giraffe here. It doesn't matter how long its neck is, because it's competing against all sorts of other clones that have sort of different but maybe similar incentives and rewards for surviving. And this is kind of the area that we're trying to operate in. So it's changing our question from a sort of evolutionary genetics, population genetics question to a more evolutionary biology ecology question. And some of the things we've been grappling with in the past years is how do you study this? Is this even a science? Can we even study this in a sort of scientific quantitative way? So I'm gonna tell you the story of a few papers that we have been working on this in this field. The first part is basically when we decided to establish this as a field, and the people who did this was the undergrad student Jeroen that Francesco mentioned. He originally started this whole thing together with Luca Messin, who came to the lab as an antibody sequencer. So Jeroen was doing imaging, Luca was doing sequencing, and you'll see how these two fall together. And Luca now is a research associate in the lab who drives most of this project. And then he was joined more recently when we moved to Rockefeller by a grad student, also from the Netherlands, like Jeroen, Arjen Schiepers, who is working with Luca on memory and other things. So the first thing we did when Luca arrived and we started to try to encapsulate this problem of the evolutionary ecological problem and try to talk about it in some sort of understandable way, we thought, well, the easiest thing that we can measure is the number of clones that are participating in response. And initially we thought, well, this might not be very interesting because the data that was around, and this was in 2014 when we did a review of the literature, basically was telling us that this was not a very interesting problem. What it said was that you had a pre-immune repertoire where you had many, many potentially responsive clones that could engage in the germinal center response. But there was a very severe bottleneck here. So once you made a germinal center, this germinal center was pauciclonal. And the number that was around that most quoted in the literature was 3 clones, that a germinal center was founded by 3 different clones. And that says, you know, the interesting part is not the germinal center, it's what happens before. How do you get from this to this? In the germinal center, then that Darwinian selection kicked in, and maybe you lost a few of these clones here and you ended up with a monoclonal germinal center, but the big bottleneck was over here. And that was sort of bad news for your broad neutralizer, because if your broad neutralizer is this yellow clone here, it wouldn't even make it to the germinal center. So you don't even have a chance to evolve it. And that's where the problem comes in. But you know, we knew that this bottleneck here existed. We had been able to document it in different systems as well. We just weren't really sure that it was that strong, that it could really get rid of almost everything and make a germinal center pauciclonal. So what we decided to do is count how many clones do you have in one germinal center at the very beginning, and ask if this here was really that strong, this bottleneck. And then we get into questions of technical questions, right? How do you count how many clones are in one germinal center if what you usually get when you immunize a mouse is you get many germinal centers in a lymph node, and then you smash them all by FACS. And what you're going to get is a compound of many germinal centers working together. So what we did use was this system that Francesco alluded to in the beginning, with this photoactivatable GFP mouse that I had made as a student in Michel's lab. And this mouse allows you to label cells using a 2-photon microscope in a precise region. And you can use this for tracking movement of cells around, which was what we did here in this paper. But there is also another use for it, which is kind of what we refer to as geotagging. If you want to do FACS and you want to know where a cell came from, what you can do is label it in the lymph node in situ. And then when you smash the whole lymph node for FACS, you'll see that the cells that got labeled here were exactly in the place that you did the labeling. Meaning you can tell that the cells that you sort on the cytometer came from this germinal center here and not from any of the other ones. So using this, we were able to generate very early germinal centers, photoactivate a single one of them, and then just sort these cells out into a plate and look at what their VDJ genes were, and tell exactly which clone they were in, and then count how many clones made it into an early germinal center. And this is what we got. We got a picture that was very different from what we had drawn in that review. That germinal centers early on, when you look at these pie charts, so let me show you one with a higher number here. For example, this, we sequenced 67 cells from one germinal center, and we got 40 different clones. And this tells you that it's certainly not 3 clones per germinal center. It's quite diverse. And this was happened over and over. These are pairs of germinal centers that we sorted from the same lymph nodes. And when they're colored here, it means there's sharing of this one clone between 2 different germinal centers. It's not relevant for this talk. But when we took these numbers here and plugged in some equations from ecology to estimate what was the total richness, the total number of clones per germinal center that we had, what we get is a number of about 100. So this says that in a single immunization, this is chicken gamma globulin. If you look at the very earliest GCs you can find and you take them out, you'll find that they are very diverse, and there's something between, you know, 50 and 200 clones per germinal center, on average about 90. Okay? And this sort of encouraged us to work a little bit more on this problem, because it's telling us that this is an interesting problem. The germinal center is not seeded in a boring way by very few clones, it's seeded by a lot. And these clones are now very different, and they're fighting with different incentives, and there's going to be some sort of dynamics with which they are going to sort themselves out. Okay? In this first paper, we did ask a simple question of, okay, they're very diverse to begin with. When do you lose diversity? And for this, we used tricks from neuroscience, these rainbow alleles, where you can fate map cells in different colors. So this Confetti allele here is downstream of this AID CRE-ERT2, which we can use tamoxifen to make these alleles select colors. And basically, what you do is you take an early germinal center that is expressing this allele, but it's not recombined yet, so you don't have a color. When you come in with tamoxifen, CRE becomes active, and you label cells stochastically in the germinal center in different colors. And this label is transmitted to the next generation of cells. So if you have a strong selective event, so such as if this purple cell gets sequenced, you select it, you end up with a single color germinal center in which all the cells are purple. Right? So we're just trying to visualize this loss of diversity over time. And this works very well. You can look at an early germinal center and early lymph node 3 days after tamoxifen, 8 days after immunization, and you see that every germinal center here looks the same color. And that's because they haven't had time to do clonal selection after tamoxifen. But if you just wait, instead of looking 3 days after tamoxifen, you look 15 days after tamoxifen, you see, you know, the output of that clonal selection at play. And you see that at the same time that you have some germinal centers that selected mostly one color and seem to have zoned in on one particular winner clone, others retain a lot of their diversity over time. And it seems to be that the way to lose diversity is to make one of these clonal bursts, and I'll show you what they look like in a minute. But this is sort of what it looks like. AID sufficient germinal centers that can do selection are these black dots. Some of them become very single colored. This is the metric we use for this over time, but most of them are not selecting as strongly and keeping their diversity. And as a comparison, these white dots here are when all the clones are the same and they don't have AID. So this is the neutral drift in the absence of selection. So Jeroen, the undergrad in the lab, managed to find a way to chop these germinal centers, these lymph nodes up in a vibratome, and then isolate a single germinal center here. And from this slice, then cut the slice in half. And from this, he managed to recover cells that he could sort. So we could actually ask the question of which cells are in this germinal center here. And what we described with this, with Luca's sequencing efforts, was this thing we call a clonal burst. That if a germinal center goes from a configuration multicolored like this to single colored like this, it's usually because one of the variants got strongly selected. And this, from the unmutated ancestor, you see that it made 1, 2, 3 mutations here, hit a node that was strongly selected. And this node was found 17 times in the 70 cells that we sequenced from here, and also a lot of its descendants, indicating that it is undergoing mutation that happened in one selective event that allowed it to take over the whole thing. And this was associated with an increase in affinity. So these kind of data allowed us to update that original model a little bit to what we think is actually going on now, which is the clonal diversity of what germinal centers do over time. The pre-immune repertoire is diverse. You select from it using antigen, but the bottleneck that allows cells to go into germinal centers is not that strong, in that it doesn't allow only a few clones in, it allows quite a lot of clones in. So tens to hundreds of clones per germinal center. And when germinal center selection kicks in, you tend to not lose that much diversity. And when you do, it is losing it for a good cause. It is you're picking a very good clone and you're making it expand a lot and leave thousands of descendants. And in all likelihood, some of these thousands are going to be better. So this does efficient affinity maturation at the expense of losing a lot of diversity. And this is kind of good news for your broad neutralizing clone that was there in the beginning, and that it will have a chance, most likely, to go into the germinal center and to evolve a little bit, which is sort of good news if you're trying to make this clone at least expand and evolve, right? Now, one problem with this is that none of these cells are actually effector cells. The cells in the germinal center don't produce a lot of antibodies, so it doesn't help your immune response to have germinal centers keep these cells. So we need to understand what the second step of this. These cells have to be able to go through another step of selection. Either they have to be selected from the germinal center directly to be a plasma cell, which is something we're working on in the lab, but we don't have a lot of data on yet. Or they can do another thing, which is exit as a memory cell. And when these memory cells are recalled, when the antigen comes in again, these can become plasma cells and make a very strong response. And this was second part of the work that Luca did, which is also published, where we try to understand what was the relationship between the clones that make it into your primary response and the clones that make it into the secondary response. Of course, following this sort of paradigm of immunology 101, which is that your second response, your secondary antibody response is much larger than the primary. And this is why we need booster vaccines and things like that, right? So what we started out trying to ask was what's the relationship between the second wave here and the first wave? And this is just a little bit of theory of how we were thinking about this. Germinal centers evolve and become higher affinity at the same time as they lose diversity, and that's what I just showed you. All throughout this history, they export memory B cells, and there has been some pretty nice work from Mark Slomczyk's lab recently showing that this is not symmetrical, that it's top heavy. So most of the export of memory cells comes from early germinal centers or even pre-germinal centers. Now, this of course creates a problem, because if most of your memory comes from here, it's going to be low affinity. So when you boost, what do you really get? Are you getting a lower affinity boost than your primary was? Or, you know, also you are exporting late germinal center B cells. So these could be better, these could respond better to the boost. And this question here is what we wanted to ask. What is the relationship between this memory pool and boost? Right? There's another thing that is interesting, and we actually kind of started via that route, which is that when you boost, you can also make germinal centers again. And these recall germinal centers were, when we started thinking about them, we thought they were most likely to contain memory B cells, because these cells are expanded and they're better. But there was also the idea that other types of cells could come in here and contribute, naive cells, for example, to this response. And of course, this recall of memory is very important, because it's the basis of many attempts to make broadly neutralizing antibodies to HIV and to flu by vaccination. This idea that you could continuously vaccinate and keep recalling the same clones over and over until you get a broad neutralizing antibody. That relies a lot on the ability of cells to re-enter. And so we wanted to understand really this ability of re-entry and how efficient is it and how might that help or not help in attempts to make this happen. So we started with a model that was pretty simple. We picked a mouse immunization with chicken gamma globulin. The only thing that's slightly different in our model from what most people do is that we wanted to make sure that the cells were memory in the second response. So we primed one foot of the mouse and boosted the other foot. And the main thing that is important about this model is that it allows us to be sure that the secondary immunization was not just the reutilization of the first germinal center. So when you do your second boost, you know that you're starting again de novo in this lymph node here that did not have a germinal center generated by this primary immunization. It also has some advantages over other models that, it's physiological transfer of cells, right? The cells from the primary response transfer themselves into the secondary response. You don't need to isolate them in arbitrary numbers and put them into the mice. It might be a closer mimic of human vaccination where you could be probably getting different lymph nodes to respond to different booster doses. What we can't do though is separate subsets of memory. We don't know where each cell comes from, and we have to do this a posteriori, and I'll show you a little bit of that. But we think this is a good model for these reasons. So one of the things we did, for example, was just ask if you generate germinal centers in the primary response and you fate map them with Rainbow, how much of this do you get in your secondary response? So we immunized the right foot pad of the mouse, this Rainbow mouse here with AID as the driver. We give tamoxifen 3 doses here. This labels about 70% of the primary response. Then we let this mouse rest, and a month later we boosted with the same thing in the other foot pad and just looked at the germinal centers here and at the plasma cells here to ask, how much of this response comes from this response. And one thing that we found surprising at that point, and some people still do even now, is that it seems like when you look at secondary germinal centers, they rely very little on the primary germinal center progeny, right? And this is very specifically for mice in a 2-dose regimen, okay? So prime and then 1 boost. When you look at the boost germinal centers in the second foot pad, what you see is mostly germinal centers that look like this. You don't even see it's a germinal center here because I'm not showing you the FDC marker. But when you look into them, you see that basically they're empty. You can see the macrophages here, but not many cells. There are several of these here. So there may be out of the 6 or 7 germinal centers in this node, only 3 of them had really substantial participation from cells that were in the primary germinal center. And if you look at the clonal distribution here, it also tells you that very few clones are doing this. There was a blue cell that made it into this one, a red cell made it into this one, and a blue cell and a purple cell, a green and purple cell made it into this one. Basically 4 dominant clones taking over the whole response, but mostly what you're getting is cells that were not involved in the primary germinal center. This is not because you didn't have secondary germinal centers. So these things here were not empty. You see that secondary immunization made a very robust response. If we use photoactivation, like I showed you before, to look at the diversity, there were many, many different cells in these germinal centers, many different clones, as diverse as the primary response. So germinal centers are being made. And it's also not because we didn't make good memory with the first foot pad, because if you look at the medulla here of the node where the plasma cells reside, you see that that's packed with cells that were fate mapped in the primary response. So the memory is being used. It's being used a lot to make plasma cells, but it's not being used too much in primary in secondary germinal centers. And this is just summarizing this here. This is AID Confetti. We use another model here that is an S1PR2 tomato that was made by Takakata and Tomokurosaki in Japan, which is more efficient. I'll just guide you through this one. At day 15 of the primary, we've labeled about 90% of cells using this CRE, meaning 90% of the primary germinal center was labeled. This doesn't go down much with time. But if you try to boost the secondary germinal center and you ask what's the percentage of cells that are labeled in the secondary, it's about 5%, meaning that 95% of the cells in a secondary germinal center did not come from a primary germinal center in this model. On the other hand, the plasmablast content of this germinal center is pretty highly dominated by cells that came from the primary. So the primary memory is there, it's just populating very well the secondary plasmablast compartment, but it's not a big fraction of the primary. And this is true across a number of prime boost models. If you prime with different adjuvants, it seems to be about the same. If you boost after a much longer time, it's about the same. If you use a different antigen like hemagglutinin, it's about the same. The only thing that could give us a little bit more participation of memory cells in secondary GCs is if you boosted the same footpad, or if you primed IP and then boosted. And both of these we think are basically, you're basically refueling germinal centers that are going on. So the first side germinal center never went away, and then you get a bit more. You get, instead of 5%, you get 10%. But in any case, we could never find a system where we can get more than 10% or so of germinal centers to be derived from memory that came from the primary germinal center. The second thing, as that graph suggested, is that the response now is pauciclonal. So if you look at the full lymph node that was boosted, and you ask how many clones were there, you start to see things like this. This was that picture I showed you. There was a big blue one, purple, some green, and a little bit of red. But basically 4 clones here dominated 75% of the response. So even though you're putting in hundreds of clones into the primary, when you boost, you're getting out something like, on average, 6 clones make up 75% of the recall germinal center response and of the plasmablast as well. So I'm going to get into that a little bit more later. One thing that is important, though, is that those cells, when they make it into germinal centers, they're fine. So they're in there. They are further mutating if you do the clonal trees and the sequencing of them. So you see that this cell here that entered the germinal center gave origin to mutants. So the cells are participating, and they are getting in there and doing what they're supposed to do. So it's saying basically that cells from one germinal center can enter the other germinal center in the secondary and evolve. They just are not doing that efficiently enough to take over the whole reaction. So only a minority of cells are doing this. And this is sort of the summary of this first few points, is that in the single boost model, the germinal centers are clonally diverse and they're very robust, but they are not made primarily of cells that were GC experienced before. They're made mostly of cells that, to the extent that we can tell, look naive. And we're saying they're at least GC naive. They do not come from the primary germinal center. They may be memory, but if they are memory, they are some strange kind of memory that is not mutated and is not very much expanded and is not class switched either. And the other thing is that what you do get from the recall into the secondary germinal center and into the secondary plasmablast is just a subset of the clones that you put in the primary. Now, we wanted to understand where is the missing diversity, because what this says is that you put in hundreds of clones into your primary germinal center, and when you boost, you get out half a dozen. Where did all that diversity go? And of course, the main place where it could be is memory. So we needed to have a system where we could reliably track memory B cells and ask, is the diversity there? So what we did was we moved to flu. Flu is interesting because, of course, it's a relevant model for humans in some extent, but it is also, there are very good tools for it. So there are very good tetramers that we can use to reliably identify cells that came, that are involved in this response, especially memory B cells. So what we did was a similar model. We immunized, we infected mice with flu, PR8 intranasal. This is the mouse-adapted H1 flu. We gave tamoxifen at days 7, 9, and 11 to label the germinal centers. Again, we get about 70% here. This is what the germinal center looks like. So you see this is at day 45, nice clonal bursts. Some retained diversity, others became more single colored. So the same picture I showed you before. But what we did was then boost the footpad of this mouse with the hemagglutinin of flu at day 45, and then just look 6 days later. And what we see is basically exactly the same story. So this here is a similar plot that I just showed you. We labeled about 70% of B cells in the primary germinal center, but when we boost, we get something like 4 or 5% if you look very early in the first forming germinal centers. And if you look a little bit later when the germinal centers are fully formed, it's even lower. So it says something like 1% of the mature germinal center B cells in the secondary are coming from the primary. But what is, of course, useful to us is that you can use tetramers to identify memory, and we can find abundant memory in the primary mediastinal node, as well as in the spleen and the secondary node that we boosted. So what we did to get an idea of how clones were working here was we just took the whole response of the mouse all over the place, from the primary lymph node to the spleen, the secondary lymph node, the bone marrow, and we sequenced everything we could find that was fate mapped and bound antigen. And we end up with these very complicated graphs that we call the clonograms that you can stare at them for hours. I'm going to walk through one kind of slowly, but there's a lot of detail here that I'm going to skip. This is one of those pie charts just flipped as a bar. Each clone is a different color. The colors will become meaningful in a minute. This is this here, this germinal center here at 45 days. You see that you have a lot of clones are dominating. You have only actually 25 clones left because there's a lot of selection. You eliminated a lot of not so good clones at this point. But when you sequence memory, you can sequence memory from the mediastinal or from the spleen or from the popliteal node that we boosted. And you'll see that here you kind of have a historical record of everything that got involved in the response. These are fate mapped hemagglutinin positive cells. And it says here that this memory compartment is much more diverse than the original germinal center compartment that you could find at day 45. And of course it is, because this has eliminated clones, and this serves as sort of a historical record of everything that went into the response. Now, what happens when you try to boost this? You make a germinal center, of course, and this germinal center was particularly poor in fate mapped cells. So only 0.8% of the fate mapped cells of the secondary germinal center cells were fate mapped. But if you look at these cells particularly, you find that they have fewish clones here expanded, but some of them can be traced back to memory that was in the same popliteal lymph node. So this kind of repeats what we saw in the immunization. But what was interesting here is that if you look at the plasmablasts in the same lymph node, they tend to be mostly clones that were not in the secondary germinal center. There seems to be a split. These clones here are clones that were mostly dominant in the primary response. But what is interesting here is that this is much less diverse than memory. So memory is very diverse. Plasmablasts are not very diverse. Here maybe, again, 4 or 5 clones make up 70% of the diversity. And this is true in the lymph node and to some extent even in the bone marrow, where we can trace the same clones again. So basically this is saying memory is very diverse. It's dividable into 2 rough compartments. One compartment that gets used, which is a pauciclonal compartment, and a big compartment full of clones that seems to not actually be used that much, which we call these sort of operationally used memory and unused memory, not very creative, but that's how we divide them up. And we can tell that there are differences between them. So used memory tends to be mostly class switched. Unused memory is IgM. Used memory is much more mutated than unused memory. Unused memory is very diverse. So, you know, of the 300 clones that we could detect out of the memory, only maybe 15 got used. I'm not going to get into this. I'm going to skip over this, but it just tells you basically that it's by clone. You know, good clones get used, bad clones don't get used, and you can measure the affinity. And so what matters is how good the clone was to start with, basically. But just summarizing all of this, when we are able to see memory, we find that most of the missing diversity lives there. So we can find all the missing diversity. And this missing diversity divides memory into a used compartment and an unused. The used compartment is only restricted. The unused is very diverse, mostly IgM, low mutations, meaning that it probably exited the germinal center early. I'm going to skip these 2 points just to show you a little model of how we think this works. So updating this, right? I told you just now that we don't have a very strong bottleneck within the germinal center. What we do have is varying degrees of selection, whereby you lose diversity over time, and diversity is lost mostly when you have these things we call winner clones that take over a germinal center, they clonal burst, they eliminate everybody else. When you ask, how does this contribute to memory? What our data shows is that most memory, that the Schlumcheck model is correct. Most memory that you get is made early. So you get this huge memory compartment. It's very diverse, lots of clones, hundreds of clones are here. But what does happen, and this is the tricky part for vaccination, is then when you boost this, the boost is very selective. So when you boost, you tend to concentrate on these winner clones that are larger, they're more mutated, they're higher affinity to begin with, and they're overrepresented in the leftover germinal centers that were generated a long time ago. And this is what is making most of your secondary plasma cell compartment. These seem to also be the ones contributing to secondary GCs, but there's a puzzle here, and a puzzle that we don't quite understand yet, which is when you look at the secondary germinal center itself, it does not seem to be dominated by these obviously memory cells, but instead it consists mostly of naive, or at least naive-like clones that look like they just joined the response, that they either left very, very early, that they were not fate mapped, or they were really naive and didn't participate in the primary response at all. And this kind of places the strong bottleneck here in the boost. And it says that this here is sort of the place that people would have to pay attention to try to get these, broadly neutralizing antibody precursors to thrive in responses, try to open this second bottleneck here and try to understand what determines the different participation of clearly memory clones and these naive-like or naive clones in the secondary response that make the experienced clones be only a minority of the secondary response. So in the last 10 minutes or so, I wanted to go into something that is a little bit more curious. It's some work that we have been doing in the past few years and that just got accepted for publication. It's waiting in line. And this was sort of an attempt that we had to go the next step and let's say, okay, we started by looking at monoclonal responses to haptens. They're very simple. We now moved on to looking at polyclonal responses to simple antigens. Now what we want to do is go to the ultimate, most difficult step, which is looking at polyclonal responses to very, very diverse stimuli. So we thought, why not the gut, right? And the reason the gut is interesting is, of course, I'm sure you've seen this slides like these many times. The gut has to juggle a whole lot of different stimuli that are happening at the same time. So there's the enormously diverse microbiota, there's the food you eat, there are pathogens that come in every now and then. And all of these come into contact with the immune system in some way or another and generate germinal centers, both in the Paris patches and in the mesenteric lymph nodes. And somehow the immune system has to be able to juggle this. So our question was, first, can the immune system make sense out of this? And second, can we make sense out of what the immune system is doing? So we started by asking, I guess, our our usual question, which is, can germinal center selection take place, especially in the midst of all of this? We know that this is true, yes. It's a very big resounding yes for vaccines and for infections. This has been shown many times. But these are very specific events where the vaccine or the infection takes over the whole response. So the response becomes monomorphic in some way. What we're more interested to know is what's happening at steady state. How is the immune system prioritizing between food and microbiota and other types of antigens? And how does that allow or not for affinity maturation? And this has been controversial. There have been many studies on this. We know, for example, that non-specific cells can participate in germinal centers. Even cells without a BCR can do that. We know that very frequently, this is work from really seminal work, I think, from Albert Bendelak's lab and Patrick Wilson's lab at Chicago, showing that a lot of the IgA that you find in the lumen is polyreactive. So it doesn't seem to be very much selected. Also, in general, the gut is generally tolerogenic, so it's not conducive to making very strong antibodies. And also the Peyer's patches in a lot of different species serve as inductive sites. So this is where diversity is generated prior to encounter with antigen. And that's true in sheep and rabbits, for example. And it's not clear that that's not the case in humans and mice. So the way we got into this was, again, with this rainbow experiment. This was part of the first paper that we published. And when we found this system worked, we wanted to say that it worked in any scenario, even when we have no idea what's going on. So we chose to look at Peyer's patches and mesenterics. And we showed, using a slightly different model here, that you can see that selection is happening in Peyer's patches sometimes, and also in mesenteric nodes sometimes. By just looking. We don't need to know when these germinal centers were generated. We don't need to know what the antigen was. We don't need to know anything to tell that there was some degree of selection happening here. Now, this was when we were at Rockefeller, moving to Rockefeller. We published this, and when we got to Rockefeller, one of actually the reasons that I really wanted to come here was that my good friend from Brazil, from grad school, Daniel Musida, has the lab just next door to mine. And he's one of the world leaders in mucosal immunology. And so when I got here, one of the first things we did was hire a joint postdoc, Carla Noasad, on the job market now, if you're interested, who we hired in order to just sort of be a probe into this world of gut germinal centers. Say, well, let's see, we start a project as simple as possible, just let's see where it leads us. So what Carla did was to just do a simple rainbow experiment, but do it in the gut. So it's even simpler than immunization. It's you don't need to do anything to a specific pathogen-free mouse to have germinal centers in the mesenterics and in the Peyer's patches. You just give tamoxifen at a random time point in the life of this mouse, and you just wait some time, and you see what happens. See, do you get, as that data I just showed you suggests, do you get single colored germinal centers? And if you do, what's that all about? So this is one of the very pretty pictures that come out of this. This is a cecal-colonic mesenteric lymph node that drains the colon, and it has many germinal centers in it. And you can see that most of them are mixed. You see a lot of this mixing even at 21 days after tamoxifen. But occasionally you can see these kinds of nicer pictures here. Like this one, for example, is very, very much dominated by blue clone. This one here seems to have YFP dominating, peppered in with other colors. And this one here is, as I mentioned before, not all of them can do clonal bursts. So most of them stay around this sort of mixed phenotype. And when you look at this, this is very interesting. You see that the normalized density score, this is how single colored a germinal center is, evolves over time rapidly. So it starts out after 1 week, not much selection has happened. 2 weeks, 3 weeks, you start to see that a lot of this, these germinal centers are actually reaching a pretty well selected state. So even in the midst of all of these stimuli, you can get quite a bit of resolution to happen. So germinal centers are able to find a winner in the middle of this mess. And what was interesting was kind of that this kind of went away over time, and the numbers are getting low here because this score is only calculated for germinal centers that have high enough density of cells in them, of colored cells. And the reason this goes away is that there's a really rapid turnover of clones in these germinal centers. So in about 2 weeks, half of the germinal center cells that were labeled are replaced with cells that weren't labeled. These are incoming clones. So these germinal centers are like treadmills. A clone comes in and evolves, and another clone comes in and evolves. But even in the face of this rapid turnover, some clones are being selected strongly enough. Some clonal bursts are strong enough that they don't allow competition to come in, and which really to us said that these are selecting very strongly, probably for a reason. So what we did was we picked out germinal centers that looked like this one. We did that slicing approach that I showed you, and we sequenced the cells from these germinal centers with winner clones in them. And we managed to see a picture that looked very similar from to what I showed you before for immunization. You see that, you know, there's progressive mutation here. This clone seems to be a clone that dominated over several steps. There was one selection event, then another one, then another one, then another one. And then at the end, you got to this sort of last clonal burst here. We found 11 mutated versions of this. And some things that looked a little bit more like the ones that I showed you, more recent clonal bursts. It says that this, you know, this event probably happened pretty recently because you have one expansion of this unmutated node here followed by the close descendants. So we can find all sorts of flavors. But what we wanted to know is this affinity maturation? It sure looks like it from the pattern of these trees, but can we show it? So what Carla did was produce the monoclonals that were encoded by these cells here, the unmutated ancestors, as well as the clonal burst points for a series of antibodies. And then she screened these antibodies for binding to the microbiota. And she found that if you screen a certain number of clones, you can find some that bind microbiota, and you do that by FACS or by ELISA. So this here is flow cytometry showing one of the antibodies. This is what it looks like by ELISA, and they have all different patterns. So some of them bind very tightly to few bacteria, some of them bind more broadly to many bacteria, but what they all had in common was that when you reverted the mutations back to the germline, they lost this binding. So even though bacterial flow cytometry is very variable depending on the input material that you have from the mice, we could see that in every experiment we did, we lost binding by reverting the antibody to its unmutated ancestor. We also did this in parallel with an ELISA of fecal bacteria. You take the fecal bacteria, fix them, plate them on a dish, and do an ELISA on it. And you can see, for example, the solid lines here are the mutated antibodies, and the dotted lines are the unmutated. So we can find that in an SPF condition, the winner clones tend to be enriched in those clones that we can demonstrably show undergo affinity maturation. We wanted to be a little bit more defined about this. We don't know what these bugs are because it's very hard to tell. So what we did was we used an intermediate model, which is what we call oligo MM12 model that comes from the groups of Kathy McCoy and Andy McPherson and others. And what this is mice that are colonized with a stable consortia that contains 12 members. And this, for example, this is the parental strain that we colonize with this, and it goes on to the F1. They transmit vertically, and that's very important. We don't want to acutely colonize mice because that leads to all sorts of strong immune response. So we wanted them to be born with this flora. And what we can do is exactly the same experiment and ask, okay, can we find winner clones against these bacteria here? And if we can we find out exactly what bacteria they bind to? And the answer is yes. This is one such example of a well-selected germinal center. We picked the unmutated ancestor and one of the mutated branches here. This is M218, we call it. And if you do a blot of the species of bacteria that we grow, we see that this 218 specifically binds to Enterococcus faecalis, and whereas these other antibodies don't. And again, if you take this Enterococcus faecalis culture and you do an ELISA against it, you see that this antibody binds to that, but it does not bind, the reverted antibody doesn't bind that well. So we can actually show that affinity maturation is happening towards commensals in a steady state condition without an infection or an immunization that we induced ourselves. So, so this was the part we set out to do. There was actually a twist in this that was very surprising, that is actually the part I liked most about it, which was on our way to making these oligo-MM12 mice, we had to go through a germ-free intermediate. And when we did that, we were surprised to see that germ-free mice actually had quite a lot of germinal centers, both in the mesenteric lymph nodes, but also in the Peyer's patches to some extent. And we were surprised, though actually we shouldn't be, because then looking back in the literature, we found that there are reports as early as 1967 showing that germ-free animals have germinal centers in them. But what we thought was, well, what are these? We don't know what they are. Germinal centers in the gut are weird. They're maybe just niche fillers, right? So maybe because you have the germinal center niche there, these cells go there and they just hang around there, or something like that. So, but we decided then to go and just do a brain experiment on them to see what happened. And what happened was very surprising to us. We saw the most single colored germinal centers that we have ever seen in any system. So this is sort of 100% red. This is 100% green. In a Peyer's patch, where it's very hard to get a single color germinal center, we saw this 100% blue, 100% red. And this was very surprising to us. If you quantify this, for example, you see that over 3 weeks, most of the germinal centers are up here in that area that I pointed to you before in the SPF. So whereas something like 10% of SPF germinal centers can come up here over this time, more than half of the germ-free ones do this. So this indicates to us that selection is very strong. It's not just fillers, right? I'll quickly go through this. I'm getting to the end of my time. We cloned the best antibodies here and looked at the winner clones. We made antibodies from them to understand how these were going. This was from one of these gigantic clonal bursts. You can see the sequence here. And we couldn't find that it binds anything. It doesn't bind food, it doesn't bind commensals, it doesn't bind autoantigens in many ways that we tried. They're not polyreactive. And that's a, well, how are they really niche fillers? Can they be just randomly selected and then once they make it to the germinal center, be selected that strongly? We don't really know what they bind to, but we can certainly tell you that that's not the case. They seem to be very, very much selected based on their B-cell receptors. And the reason we know this is that we started looking at these and we started to see these clones pop up all the time that were always the same. So this is a V147. And almost every time we looked at winner germinal centers in mice, we could start to see these this exact clone or similar clonotypes pop up over and over and over. So we have two clonotypes that were our favorites. This is 147 ARGSNI, we call it, which has these 6 amino acids at the beginning of the CDR3. This is a very strict one, V112 AREG-FA, that has this 7 amino acid CDR3. And these are pretty rare among the naive population, but when you look at germinal centers, they're pretty common. So this one here, for example, 20% of our winner clones seem to have this across multiple mice. So certainly it's not a niche filling by random clones. This is very heavily selected. Very interesting also, Fred Alt had a paper that just came out in Nature that was sort of contemporary with ours, but came out much earlier, where he shows these two exact clones popping up in Boston in his germ-free. So it's not even something specific of our colony. This is all over the place. We can quantify this better. We can see that these clones are present all over the place in germ-free mice. Also, oligo-MM12 is not strong enough to eliminate them. And often, and occasionally we can even see them in SPF mice as well. This is using a much wider cold stream, non-biased system. That also shows you that there's a lot of clonal sharing in addition to these clones. So we can identify at least 15 clones that have these properties being all over the place, public clonotypes in mice. So to summarize this, the first part, gut-associated germinal centers select winner clones well in spite of all the antigens that they're exposed to. Affinity maturation is detectable in steady state germinal centers to commensal. So polyreactivity does, of course, exist, but it's not the only game in town. There is some degree of affinity maturation towards commensals. We don't know how much this contributes to gut IgA, and this is something we're looking at. Selection in germ-free mice is greatly accelerated. It converges on these very stereotyped clones that we can find all over the place. And the conclusion for this is that in germ-free, the germinal centers are not just made of niche fillers, but they're instead very heavily selected based on the B-cell receptor. And a lot of what we want to do and what Carla wants to do is continue to try to understand what these clones are all about. So just to finish this is our lab. People that have done most of the work I've acknowledged already. Jeroen is not here. He started all of this when we were at Whitehead, but Luca has been with us all this time, and he is the driver of the whole clonal dynamics branch of the lab. Arjen is the grad student who is a co-first author with Luca on this memory recall paper. Carla is driving a lot of the secondary of the gut work together with Luca, who has done a lot of sequencing, and Tiago is our bioinformatician who did a lot of the second germinal center analysis that I showed you, a lot of the analyses that are in here. We've had lots of collaborators who've given us mice, have given us reagents. We couldn't do our work without them, and also a lot of help from modelers that help us understand our make our trees and interpret them. And also, of course, the second part was a very close collaboration with Daniel Musida's lab here at Rockefeller. And these are our sources of funding. So I'll leave you with that. Thank you for listening, and I'll be happy to take any questions.
All right, Gabriel, thank you so much. That was outstanding, to say the least. Really fascinating, really beautifully done.
Sorry for cramming so much information. I have to streamline this, I think.
You did a great job. We have a lot of questions and unfortunately not that much time. So we'll start with a few of them, and then for everybody in which I will not manage to ask the question to Gabriel directly, I will pass your question to him via email alongside with your coordinates to, to be contacted about the answer of your question. I apologize for that, but
sorry, yeah, it's probably my fault for making this too long.
Well, that was definitely fascinating, though.
I've lost my practice with giving talks.
So we have one question from Lisa Wager, our newest recruit, asking what are your thoughts on clonal competition in prime boost schemes, wherein the boost has some shared but also unique epitopes. For example, as it happens with seasonal flu drift variant.
Yeah. This is a very interesting question. When we do boost with a different strain as we primed in flu, we get pretty much the same result. Very few cells come from the primary and the secondary. That's sort of expected, I guess. But we had this idea that the unused memory might be all over the non-shared epitopes or the less conserved epitopes, but it didn't seem to pan out. We see about the same thing. As I was telling Lisa earlier, this is a mouse with a 1' and 1 boost. So the history of exposure is very short. So we don't have this ability to build up memory over decades like human studies have. And I think, that the contribution of memory is going to increase as memory increases. And I think we've seen some beautiful work from Ali Elabedi's lab in human now showing this variation, how just came out recently, like some human recruit very few memory and more naive, and other humans subjects recruit lots of memory and less naive. And I fear that the mouse might not have the lifespan for us to do these experiments repetitively properly, so that we can get the idea of how to generate cross-reactive memory and recall it later, as a human would do.
Okay, so one further question is from one of our graduate students here, Zach. He is asking, is it possible to do epitope mapping to see if epitope accessibility affect the likelihood of clonal B cell dominance or recalling to germinal center for maturation, for further maturation?
Yeah, we are starting to try to work that out in flu. We have good tools for that. From John Udell made some very nice mutants that have lots of epitopes knocked out. We're trying to get there. So far, what we can see in flu is an obsessive dominance of the head over the stem, but I think that's no surprise. But whether this comes from competition in the germinal center or not is we haven't been able to tell yet, but it could be that that's the case.
Okay, then Scott, another of our graduate students, asked whether the starting diversity of B cell clones actually matters in terms of the final result after selection. So do you have any, variability in the starting population of BCRs? And if so, does it matter for selection
protection?
Center?
For selection or for protection?
For selection.
For selection.
For final finality, yeah.
Yeah, we don't know that. We are trying to model this slowly with some less diverse systems. I mean, we can see good selection in B18 monoclonal NP specific. So when everything is monoclonal, you still select very well. The best cells, and you also do this thing where some germinal centers select and others don't. When you do CGG, which is very diverse, or even in the gut, you seem to get the same. I don't have a good feeling yet for whether starting diverse means you get to a better point at the end or not. My feeling would be that no, probably you will get to good antibodies regardless, but that's just guessing at this point.
Get you. Then Michael Buchmeyer asks, basically the relationship between the memory you get in the first immunization or exposure to antigen and the secondary infection that is completely unrelated. So do they talk to each other? Have you ever observed a clone that selects in the first reaction to show up in the second one, or it is based
to a different pathogen, you're saying? To a completely different pathogen?
Yeah, exactly.
Yeah, that's a really good question. You know what? One of my favorite experiments in human is from the head of Waterman's lab, who showed that she gave malaria sporozoites irradiated to a bunch of Germans who had never seen malaria before because they're Germans. Yeah. And they responded with memory. Whoa. So their memory plasmablasts came up in the blood. Whether this is a malaria-specific thing or whether they're cross-reactive or whether, this, I don't know. But it's interesting to think that when you make all of this unused memory, you're sort of pre-licensing these cells to become plasmablasts if they are the only game in town. So that's a really interesting thought. We don't have evidence for it yet. It's very hard to model these things, but it is a very interesting possibility, I think.
Okay. I'll ask probably one last question from Adam Feig. He asks that given that autoimmune germinal center and foreign antigen-driven germinal center clonally evolve at comparable rates, do you know about the dynamics of the MBCs generated during chronic persistent antigen exposure and their ability to re-enter the germinal center? And BCs, I suppose it's memory B cells.
Yeah. We have done some experiments with chronic infection. These are not published yet, but they're going to be coming at some point. We infect 2 mice chronically. We fate map 1, and we do parabiosis.
Yeah.
And we do not see the cells from one enter the other during the chronic infection.
Excellent.
Admittedly, a mouse model of a chronic infection with hepatitis C, which is not a particularly strong, we're trying to see if we can do this with LCMV chronic infection as well. But we don't see that happening. We also don't see gut exchange in parabiosis. So cells from fate mapped in one mouse don't end up in the gut of the other mouse.
That's
so
awesome.
It doesn't seem like it, but it's low resolution so far. We didn't have to do it more to say something really about the gut.
Perfect. There are more questions, but I will actually ask them to you via email so that we can move on to the next session.
I'm actually sorry. I just realized now that I haven't been giving talks for so long that I'm not supposed to end at the hour. I'm supposed to end 10 minutes before the hour. So I was timing myself at the hour.
Don't worry at all.
Sorry, guys, those questions I can't answer.
And now please join the session.
I just want to say thank you on behalf of the whole.
And all students, please join the session with Gabriel, and see you there. Thanks, everybody.
There's a different Zoom setup.
All right.
Sorry, Eric.